so much.
Any questions?
Yes, what's the evidence
Yeah.
that EGR1 can really do that? You know, this is all correlation, so where's the causal evidence?
So this is predict, right? EGR1 is predict in the beginning, but we need to provide evidence. This is a very good point. We start with a lot of computational work. And in the end, we need to prove on the bench. So I trust bench. We're doing a chip sequencing. Basically, you using antibody pull down this protein, whatever DNA bind. And we check the methylation change through the development. And you see that this methylation change only occurring in neurons. And during the development, it gradually lost the methylation. So that is how it happened. And this protein is very well known. People know that for many years. You can find thousands of papers published for this protein, but they just don't know it changed your methylation. Okay? Did that make sense? I can show you more data.
Actually, you just don't have to just believe him because he says to believe him. You can read his papers.
Oh, yeah. And the paper is pretty much done. Yeah, I finished the paper. Yeah, thank you.
Any other questions?
Hey, David. I have another question. So I know that some of your work has to do with epigenetics in early development, and I was wondering whether you can, I'm very interested in how this, environmental factors might influence at the epigenetic level. So how early these changes happen?
Okay, very good question. So a lot of detail I need to bring it up. EGR1 protein expressed low in the fetal, and then after baby born, it starts to go up. So it's critical for the postnatal early time, early 3 years. It jump up. And now this protein, what it does that when I talk to you now, you start to think, right? And in millisecond, this gene is go up. And then it go to nuclear, do something there. And then 1 hour, it will drop, go back, calm down, quiet. So this process is very quick. And this happening, I believe, personal belief, is a postnatal life. And the beginning of our life period is so important. So many long-term memory things established at that long time. But I know the marker will switch are, but understanding the switch is going to take me another, I don't know how many years. So, hope answer your question somehow.
Great. So this is a non-EGF question, but for the rest of the panel, because I know the MIS division has a lot of focus in translational diagnostics. And so it's becoming, more and more evident that these algorithms tend to have different benchmarks, right? And so where do you think your algorithms, or where do you think the standardization process is really going to go in the next 5 to 10 years? Because this is something that I think will be heavily regulated because it brings in human health. So.
Well, let me start this. It's not on. The algorithms to date have been built really for a particular purpose, to assemble the genomes when there was no genome 10 years ago, to reassemble genomes on top of it. And really, the kind of efforts has been to do things like, well, let's call the most number of SNPs or most number of variants that we have, as opposed to high quality variants, things that, that are potentially clinically valuable. And so the effort now is really turning to trying to develop technologies and techniques to not just call variants, but to also call robust variants that have some clinical implications, that can be exploited, that are actionable. So you're going to actually see a lot more work in the future to come up with things that are actionable. So that, and I can tell you, if you're actually trying to get anything into the clinic or into an investor to get it into the clinic, the word actionable is an essential part of that. And so I think that that there's going to be less kind of academic freedom to just call as many things as you want, as opposed to calling things that are high quality. David, you probably got comments on this.
Yeah, sure. I have. Sorry. Yeah. Oh, if it's on, I didn't realize. So I have a response. I think right now the whole genomics market, whether you're in the academic or in the industry sector, is very fragmented. It's like in the 90s when people were buying video cards and motherboards and assembling computers, and then those were consolidated and replaced with verticals. So I think if you look, for example, at what the FDA is doing now that it's pushing for LDT regulation. And they're already telling you, you don't have to guess what they're expecting in the next 5 years. And in CLIA and CAP, what you're going to find is that more and more, and this has already happened, like in CLIA and CAP labs that I've been part of, they don't really care what algorithm you're using, and they don't care what sequence you're using. What you're doing is you're offering a test, which is a vertical that integrates the sequencing, whatever you do with the data, and the reporting and annotation and everything else. And then you benchmark that against tools like GCAT, which has actually been cited as a way to do FDA and CLIA and CAP validation, against other truth sets, against the CAP. Like CAP is now going to be sending around kind of like test samples. You run your thing and you see if it matches up. And so I think the algorithm becomes like one tiny piece. It's irrelevant to optimize an algorithm because it's heavily coupled to how you do the sequencing, what platform you use, even the kind of input DNA going in. So I think those details will kind of fade. And then what's really going to matter is just if you advertise that you can pull out this amount of information, that you can pull it out. It's the same problem 23andMe faced and everyone else is facing it. It doesn't matter if you're sequencing on an array or whatever you're doing. They're just going to want to know that your actual full stack of technology produces something that is what you're advertising it produces. So I think that's what's going to happen. You won't see any more companies that are offering bioinformatics service or sequencing. There'll just be people that offer full tests.
Okay, great. Thank you for the thorough answer. One question from Bishop, and then we got a...
Well, I was out for a couple of minutes. I don't know whether anybody asked or not. This is for Mahalik. The question is, you are calling it antagonism, and you say ancient. How far can you go down to see where it started? Without mitochondria?
I guess the question is about genomic conflicts. Mm-hmm. They're very ancient. It refers mostly to eukaryotic genomes, mm-hmm. Where you have maternal and paternal mm-hmm. Genes, and it's especially an application to, it applies to organisms that have like parental care. There's some intimate contact between offspring and mother. So there is there is a conflict between, first of all, mother and offspring, which is quite known, but within the genome also there is a conflict between maternal and paternal genes. And also, you mentioned mitochondria. Yes, mitochondria are a part of the conflict because they come from mother only, so it's maternal inheritance. And there are some well-described examples of conflict between mitochondrial and nuclear genes. But this one that I described is just, apparently it's all within ribosomal DNA, which also the structure of the complex has a hallmark of genetic conflicts because of its repetitive nature. There are multiple copies of the genes, so there is some redundancy that doesn't make any sense. And I think the easiest explanation is that the whole system is driven by some ancient genetic conflicts.
Okay, great. Well, we can give a round of applause to the MIS division for a very nice presentation.